Thank you to everyone for joining the 2025 Cell and Molecular Physiology section Hugh Davson Distinguished Lectureship webinar. I just like to take one moment to let everyone know that this webinar is being recorded. Our featured lecturer, Bonnie Blazer-Yost, and we have a disclaimer before we begin. Photo or video capture of any scientific or professional development presentation in whole or part without the express permission of the presenter is prohibited. AI transcription is also prohibited. You must receive express permission from a presenter before sharing the contents of their presentations on social media or elsewhere. Please respect these requests for privacy as a professional courtesy. Thank you all for attending.
Okay, great. So my name is Dr. Matt Triolo. I'm a postdoctoral fellow at the University of Ottawa, and I'm the junior trainee representative for the cell section of the APS. And I'll be co-chairing this event with Dr. Sandrine Pierre, who is a professor at Marshall University, and she's the previous chair of the cell and molecular physiology section of the APS. And before I pass things off, I just want to briefly highlight the Davson Award, which is a distinguished lectureship in the cell and molecular physiology section. And it's awarded to a scientist who's made major contributions in the scientific areas covered by the cell section in honor of Dr. Hugh Davson. And the awardee is nominated and meticulously selected by our steering committee. On that note, I'll pass things off to Dr. Pierre, who has a few words to say prior to us beginning.
Thank you very much, and good morning or evening to you all joining us. Thank you for this event. It's a pleasure to share that the 2025 Davson Distinguished Lectureship Award recipient is Dr. Bonnie Blazer-Yost, who is a professor in the biology department at Indiana University. Dr. Blazer-Yost earned her PhD from the University of Pennsylvania and completed a postdoctoral fellowship at Cambridge University. As many of you know, her work as a leading cell transport physiologist and an esteemed mentor to many has earned her numerous awards, including the Outstanding Research Award for Tenure Faculty at Indiana University, Purdue University at Indianapolis, the H.H. Singh Lectureship of the APS, as well as the induction as a Fellow of the American Physiological Society. Her research, her lab, specializes in the regulation of epithelial ion transport mechanisms that are essential for salt and water homeostasis. Over the years, her laboratory has conducted extensive studies to examine the role of the kidney in the development of hypertension in the context of metabolic disease. Her current research focuses on developing drugs to treat hydrocephalus, aiming to address both acute and chronic forms of the condition and advance to clinical trial. As you maybe are aware, the Davson Award per se was presented to Bonnie during our annual section banquet at the APS, and this is a few pictures of the event that was in Baltimore, of course, last month. At the summit, as the Davson Award recipient, she also engaged with members and trainees in multiple section events, including the trainee breakfast and the Davson event. I think the next slide, we have pictures of that event where we showcase presentation by the new investigator and research recognition awards. I don't know if the next slide is available to illustrate. Yeah, so those were research awards, and Bonnie chaired that session, which was a great success. Now, today is the distinguished lectureship in the form of a webinar, which allows the Davson Award to reach many across the world. And as many of you know, since the Davson seminal contributions, we have learned much about the role of the cerebrospinal fluid in health and disease. Then it is highly fitted that Dr. Blazer-Yost's Davson lecture entitled Cerebrospinal Fluid Production by the Choroid Plexus Epithelium will focus on the role of electrolyte channels, pumps, and transporters in the production in CSF. And the emphasis will be on understanding and treating hydrocephalus. Just before give the floor to Dr. Blazer-Yost, just a reminder to please use the Q&A function rather than the chat for any question that you may have for her. Dr. Blazer-Yost, congratulations again on the award, and thank you.
Thank you so much, Sandrine and Matthew, for that introduction. I'm going to turn my screen off, or my video off. Can you see the screen that has the title on?
Yes, we can.
Okay, thank you very much. I would like to express my gratitude not only to for the introduction, but also for the honor of being able to present this lecture. I've always considered myself a physiologist, and the cell section of APS has always been my scientific home, and that goes back decades. I've enjoyed interaction with colleagues over the years, and so I am particularly happy to be giving this lecture. Before I start into our data, I would like to consider the luminary for whom this lectureship is named, and that is Hugh Davson. The picture of Hugh Davson, which is shown on the slide, was taken during an interview by the Physiology Society of Great Britain. And if you want to read that, it's listed on the bottom of the slide. It was conducted in 1995, a year before his death. And when he was asked during that interview about the recognition that he was most proud of in his career, what he replied was the APS lectureship, which had been established in 1993. During his career, he was known for the Davson-Danieli model of the plasma membrane. That was the first model that incorporated proteins as part of the lipid bilayer, albeit they weren't incorporated into the lipid bilayer, but just as part of that bilayer. That was in 1935, very early in his career. That, of course, was supplanted later in the 1970s by the Singer-Nicholson fluid mosaic model that is in most textbooks today. But his research was predominantly on the production dynamics and composition of the ocular and cerebrospinal fluids. And he made many contributions over the years to that field. He also was the writer and editor of well-respected textbooks at the time of general physiology, of physiology of the eye, and my favorite, physiology of the ocular and cerebrospinal fluids. And I was lucky enough to obtain the first edition of that latter text, written in 1956. And in preparation for this lecture, read some of that textbook. It was fascinating to read, especially when one is considering the secretion of these fluids, of the ocular and cerebrospinal fluid, without a knowledge of how things are transported across an epithelial cell. This was before Using's elucidation of polarized transport. It was before the sodium-potassium ATPase driving energy for movement across cells. And the textbook is written in a way that defines all of the experiments that contributed to what was known at the time. And then, as an aside, Dr. Davson was not shy about adding rather blunt editorial comments into the footnotes. So it was a rather fascinating read, and I think we are really indebted to all of his research over the years, particularly those of us who are looking at the production of cerebrospinal fluid. The other acknowledgement that I would like to do up front is the people that have been involved in the research over the years and are currently involved in the ongoing research. I have the pleasure of being the director of the Hydrocephalus Research Center at Indiana University, and most of the current members are shown in this picture. It's a great group of very committed scientists and a real joy to work with. The research center is relatively new, and since I'm going to be giving an overview lecture on how we got to this point in our studies, I will not have time to talk about the new experiments that the members are doing. If I have time at the end, I will summarize some of our recent directions. I also want to acknowledge the previous members of the lab whose research I will be talking about, and our long-term ongoing collaborators listed here, as well as our funding from various sources. So for those of you who aren't as familiar with the field of hydrocephalus, what hydrocephalus is a dysregulation of CSF balance that causes an increase in the fluid compartment of the brain, in the ventricles. The ventricles are shown in this cartoon in blue, and obviously the expansion of the cerebrospinal fluid, as you see on the right, is going to have profound effects on the rest of the brain. Now, when one thinks about hydrocephalus, I think most people think about the pediatric form of hydrocephalus, which is shown in this slide. Because as the cerebrospinal fluid builds up in the brain for whatever reason, if the skull is not ossified, as it isn't in neonates, then the head can expand. And you see here two rather horrendous examples of that. But I do want to point out that hydrocephalus is not confined to the pediatric population. It can occur at any age. One thing that has been underdiagnosed over the years and is just really coming to the fore in diagnosis is idiopathic normal pressure hydrocephalus, which is a form that shows up in the elderly population. It's often mistaken for Alzheimer's or Parkinson's, which is unfortunate because while the symptoms are similar, unlike those diseases, NPH can be surgically treated in the population, and if caught early enough, can actually reverse some of the symptoms. So hydrocephalus has multiple causes. It can arise from an infection, stroke, traumatic brain injury, tumors, which block the flow of the cerebrospinal fluid, or more rarely, genetic malformations. And here is just the schematic showing the estimated stats for the various populations. And as you can see, hydrocephalus in the elderly is indeed the most prevalent form of hydrocephalus, followed by the pediatric form, which most people are familiar with. If we look at the whole world, you'll see that as prevalent as hydrocephalus is in westernized countries, it is even more prevalent in third world countries. And that is predominantly because of the post-infectious hydrocephalus in the neonatal population, which is a huge problem in third world countries and very difficult to treat. Speaking of treatment, I want to underline the fact that there are absolutely no drugs to treat hydrocephalus. There are many labs, ours included, who are working in that direction, but to date, there are no drugs that are effective in treating hydrocephalus. So the only treatments are surgical. And the most common of the surgical treatments is the implantation of a shunt, which is implanted into the ventricles to drain the excess fluid. And while this is a life-saving surgical intervention, it is imperfect. The shunts very often require revisions. In fact, the best statistics tell us that the average half-life of a shunt in the pediatric population is about 2 years. So if one talks to patients who had hydrocephalus as children and still have shunts, they may have undergone 20 or more surgeries before they reach adulthood, and these are brain surgeries. Another more recent treatment that is applicable in some cases, not all cases of hydrocephalus, is to create an opening, a hole in the floor of the third ventricle, as is shown in the second diagram. This, again, leads to drainage of the excess cerebrospinal fluid, and this procedure is often combined with cauterization of the choroid plexus, or some of the choroid plexus, which is the tissue that produces the cerebrospinal fluid. So let's talk then a little bit about cerebrospinal fluid. Adult humans produce about 0.5 liter a day. And the buildup that forms hydrocephalus can come from many causes. There can be a blockage of CSF flow. Certainly that is the case in many tumors that are in the brain. It can be an underabsorption of CSF, because if the brain is producing 0.5 liter of CSF a day, then that much has to be reabsorbed. Or it can come from overproduction of CSF. In our research group, we're taking the rather simplistic approach, saying that regardless of the cause of the hydrocephalus, if we could control the production of CSF with drugs on an as-needed basis, this may be a helpful addition to treatment. And that is what our research is really focused on, understanding CSF production and identifying potential targets for drug development. So how is the CSF produced? Well, it is produced by an entity called the choroid plexus. The choroid plexus is found in the lateral ventricles, shown here in the purple, in the third ventricle, kind of the pink color, and in the fourth ventricle. The choroid plexus is a rather complex structure. In the insert here, you see the choroid plexus from the rat model that I'm going to be talking about later. You can see that it's a very tufted structure. It is arguably the most secretory tissue in the body. The only tissue that I'm aware of that may be more secretory is the intestinal tract during secretory diarrhea. This little tissue is amazing in the amount of CSF it produces. The next 3 slides are taken from a review article by my friends and colleagues, Helle Damker and Jeppe Pretorius. While this version of their review is over 10 years old, I really like the graphics, so I am using those here to show the makeup of the choroid plexus. So what we're looking at in this diagram is the blue area is the cerebrospinal fluid in the ventricles. And the ventricles are lined by ependymal cells, shown here. And at the outpocketings of the choroid plexus, which you see here, the cells of the choroid plexus are contiguous with the ependymal cells, but their structure changes. They become more cuboidal. They become much less permeable. They are truly a barrier epithelial cell, separating the vasculature of the choroid plexus from the CSF. The vasculature, speaking of the vasculature shown here of the choroid plexus, this is very unusual in the brain because it's a fenestrated capillary system. So as a fenestrated capillary, it's leaky and you have a good deal of filtrate formed within that area of the brain. The epithelial cells, the choroid plexus epithelial cells, control the movement of the filtrate into the CSF. But I have to underscore, and it's extremely important, that CSF is not a plasma filtrate. The composition of the CSF, both electrolyte, fluid, and some other components, resides in the epithelial cells. So those are the cells that we are most interested in when we talk about CSF production. This is a schematic also from the same review that outlines some of the channels, transporters, and pumps that are found in the choroid plexus epithelial cells. On the apical side of the cells, facing the CSF, you have a number of transporters of various descriptions. Likewise, you have separate transporters on the basolateral side facing the filtrate. And of course, it is these transporters and others that may not be listed here that control the composition and amount of the CSF. Couple things I want to point out because they'll become important in a bit is that this this type of epithelial cell is a little bit different than most epithelial cells. The sodium potassium ATPase, the pump, is on the apical membrane as opposed to the basolateral membrane of most other epithelial cells. Sodium potassium, the NKCC1, sodium potassium 2-chloride transporter, is also on the apical membrane. Also shown in this slide is the fact that we know very little about the intracellular regulators that govern the activity of these channels, transporters, and pumps. Shown in this particular diagram is carbonic anhydrase as an intracellular regulator, which underscores the importance of bicarbonate in CSF production. Now, obviously, in the last 12 years or so, we have understood a little bit more about the intracellular regulation of CSF production, but there's still a lot of holes in our knowledge about this. So we don't know which of those myriad transporters are crucial, which are rate limiting. What we do know is that the final outcome is a situation where the CSF is quite different from the plasma filtrate. The CSF is higher in chloride, lower in potassium, The pH is lower in the CSF. And I don't mean to indicate that this situation is really this static, because the composition of the CSF changes on a diurnal basis, particularly potassium. Concentrations change. They may even have some control over wake-sleep cycles. So very interesting, these differences and how they are controlled. So with that background, I'd like to give you an overview of our studies in the field. My foray into CSF production started rather serendipitously. As some of you in the audience know, I was a renal physiologist for most of my career, and I was studying PKD, polycystic kidney disease, when at an experimental biology poster session, a colleague asked whether we would use some of our animal models to look at an agonist of a transport protein called transient receptor potential vanilloid 4, TRPV4. Everything we knew about PKD, polycystic kidney disease, suggested that activation of TRPV4 would decrease cyst production. Well, because our studies were not funded, we chose a rapidly progressing model of PKD. Certainly not the best model, because PKD is a very slowly progressing model. And in the rapidly progressing model, we didn't see an effect on the of TRPV4 agonists on the kidney. Others have gone on subsequently to show that TRPV4 agonists do have an effect in the kidney on a more applicable model. But in our case, we noticed a different effect. We noticed that the animals we were treating were dying early. And which is saying something, because in this model, the animals only last to about postnatal day 21. They have both polycystic kidney disease and they have hydrocephalus. Well, a very acute graduate student who was taking care of the animals said, you know, I think their heads are bigger. I think their hydrocephalus is worse. We followed that up with experiments using calipers to measure the head of these animals. And in fact, the TRPV4 antagonists were making the hydrocephalus worse. And since there were hydrocephalus antagonists available that had been used in animals, we treated the animals and it ameliorated the hydrocephalus. So here's an introduction to our animal model. They were originally called the polycystic kidney disease animal, Wistar polycystic kidney disease, WPK. We now know them as transmembrane 67 rat model. They have a single point mutation in a protein called meclin, and this mutation is orthologous to the cause of Meckel-Gruber syndrome 3 in humans, a ciliopathy. So the homozygous animals have both polycystic kidney disease and hydrocephalus. You can see that as litter mates, the affected animals are smaller. They have domed heads. They have sunsetting eyes because of their hydrocephalus. If one does an MRI of their brain, we're now comparing on the right, the hydrocephalic lateral ventricles to the wild type lateral ventricles. So you can see there's a huge increase in the ventricle size in the affected animals. So the data I'm going to show you in a moment, I want to go over our experimental protocol. What we did for this study was we did a pre-treatment MRI of the animals at day 7. As I mentioned earlier, these animals don't survive past weaning, so we only use them when they're healthy enough to be feeding and growing. And so we'd use them between 0 and postnatal day 15. So postnatal day 7, we do an MRI. We then treat them daily with a TRPV4 antagonist. to day 14 and then do a post treatment MRI at day 15. The data I'm going to show you are expressed as the difference in the lateral ventricle volume between day 7 and day 15. So if we look first at the wild type animals using the vehicle or the TRPV4 antagonist, you can see that there is an increase in ventricle size from day 7 to day 15. You would expect that because these animals are rapidly growing. What we were very pleased about was that the TRPV4 antagonist did not decrease ventricle size. That's extremely important because you don't want to be changing normal ventricle size. Now, in the affected animals, in homozygous animals, you can see the vehicle-treated animals have a huge increase in ventricular volume for these tiny little animals. Then that increase is completely ameliorated by treatment with the TRPV4 antagonist. So what is TRPV4? Well, it's a very interesting channel. It's a non-selective cation channel. When it's activated, it allows the influx of both calcium and sodium into the cells. In subsequent experiments, which I will show you, we do know that TRPV4 is found in the choroid plexus. It is on the apical membrane facing the CSF, as shown in this slide. I do have to point out that we redrew the slide, only putting in the transporters that we are most interested in studying. So we've simplified the original version of the choroid plexus transporter story. TRPV4 is activated by stress pathways. That can be osmotic stress, mechanical stress, temperature, inflammation, arachidonic acid metabolites. That list is taken from the literature, and from the literature looking at TRPV4 in many different tissues. But as you can see, the activation is always by stress pathways. So the question became, is TRPV4 active in our choroid plexus cells? And the answer to that is yes, it is. What you're looking at here are freshly isolated pieces of choroid plexus from our rat model, wild type rat model. In the picture shown in panel A, what we did was incubated the tissue with FLO4, which is a calcium indicator. We allowed the FLO4 to permeate into the cells, and then as a positive control, we added ionomycin, a calcium ionophore. And as the calcium flowed into the cells, you can see the FLO4 lit up. Now, instead of ionomycin, if one adds a TRPV4 agonist to the cells, you can see that there is a huge increase in calcium. One thing that is reported in the choroid plexus cells, and in the case of TRPV4, is that sodium also flows through the TRPV4. So now we're doing, in panels B, we're doing the same experiment using a marker of sodium influx, corona green. Our positive control is nystatin, and as I'm sure you're well aware, nystatin allows sodium and other electrolytes to flow into the cell. But when you add nystatin, you see this increase in sodium influx. If you add the TRPV4 agonist, you see a similar increase in sodium influx. So yes, the TRPV4 is present and active in those cells. So just to summarize the two major points so far is that there are a lot of secondary effects of TRPV4. If you increase the intracellular calcium, you're going to secondarily increase transepithelial ion flux through calcium-sensitive channels, particularly chloride and potassium channels. Also, the increase in the intracellular sodium has the potential to change electrochemical driving forces for several transporters, particularly those found on the apical membrane. So our TRPV4 antagonists decrease hydrocephalus in a genetic rat model. TRPV4 is in the choroid plexus epithelial cells and appears to be functional. So now the question is, how is TRPV4 working in these choroid plexus epithelial cells? In order to look at that question, given where the choroid plexus is in the body, we decided that we really needed a continuous cell line. And I scoured the literature for cell lines that may be applicable to the types of studies that we do, which include electrophysiology. And of all the papers that I could find, there was only papers from one group, and that is the groups of Professors Schwerk and Schroden in Heidelberg, Germany. And they had two different cell lines, a porcine and a human cell line. And these cell lines had the characteristics that we felt were important for future studies. Namely, that they formed a moderately high resistance epithelium, so a barrier epithelium like the cells do in vivo, and that they had the polarized distribution of transporters that matched those, again, found in vivo. These professors and their team have become valued colleagues and collaborators. They shared both cell lines with us. I'm going to be talking about the human choroid plexus cell line. Grows predominantly as a monolayer. It has a moderate transepithelial resistance. We only use cells where the monolayer is above 300 ohm centimeters squared. And it shows this cell line shows the endogenous expression of the transporters that we were interested in studying. We added to the characterization that had already been done in this cell line. We looked at choroid plexus epithelial cell markers, cell lineage markers. We looked at junctional complex proteins. Incidentally, these are all PCR graphs that you're looking at. We found that the native transporters that we were interested were there, including aquaporin 1, which is found in the choroid plexus cells. So that brings me to the technique that we use in our cell culture study, and that is using chamber electrophysiology. I'm going to go over it just very briefly. We grow the cells in 6-well transwells, which you see here. The cells are growing on a permeable support in these transwells. We grow them until they are electrically tight, until they have resistances above 300 ohm centimeters squared. At that point, we cut out the filter and put it in what is called a using chamber. In the using chamber, you have ports for electrodes, and those are shown by these little green circles. The electrodes that are closest to the cell, one on each side, measure the spontaneous potential difference across the epithelium. That is connected to a current voltage clamp with the other electrodes that are here, and the potential difference is clamped to 0. Now, when that potential difference is clamped at 0, you can, the short circuit current is a measure of net electrolyte flux. So it's all the positive and negative charges moving across the epithelium in both directions. And let me underscore the fact it's moving from one side of the cell to another. What we cannot measure using this technique is flow into the cell. You can only measure flow from one side of the cell to another. And it is the net flow of all of the ions. So to interpret the graphs that I am about to show you, a couple things. The short circuit current, net ion transport, if the current moves, or the net current is in a negative direction, that is indicative of either cation secretion or anion absorption. A positive is the opposite. During the experiment, we can put non-zero voltage pulses across the tissue. And when we do that, there's a change in the short circuit current that is commensurate with the voltage pulse. So using Ohm's law, we can, which is V voltage equals current times resistance, IR, we can measure the transepithelial resistance. The inverse of the transepithelial resistance is conductance. So conductance is a measure of transepithelial permeability, and that is permeability either through the junctional complexes or through the cell itself. So now I'm going to show you some data. The first graph here is the short circuit current. At time 0, we're going to add a TRPV4 agonist to all of the cells. So let me walk you through this. And I want to call your attention first to the black line. And this is a pure TRPV4 agonist trace. So you add the TRPV4 agonist, you see an immediate increase in short circuit current, and that increase is followed by a decrease back past the basal current. So there's always a level of basal current in epithelial cells, and this dips below the basal current. If 5 minutes before the addition of the TRPV4 agonist, we add an antagonist to the cell, and that's shown in the pink, we block all of the subsequent response to the TRPV4 agonist. And probably the most interesting component of this experiment is if we add a TRPV4 agonist, and that's shown in the teal here, and 5 minutes after the addition of agonist, we add an antagonist, you see that the current goes back to about baseline levels. Now let's look at the conductance. In the black line, again, we have the TRPV4 agonist only, and the conductance goes up and it stays up. So the permeability of the cells has gone up. As an electrophysiologist, this degree of conductance change is bigger than I have ever seen in renal cells, in intestinal cells, and in lung cells. There is a huge difference in conductance. Now, again, that could be transport through the junctional complexes, it could be breakdown of the junctional complexes, or it could be transport across the cells. As, again, an electrophysiologist who's done a lot of this, when I see a conductance change like that, my first thought is something's wrong. The compound we're using is toxic. Well, it turns out that is not the case. It's just that the cells are transporting so much material that the conductance has stayed high. And how do we know this? Well, that's why we did the experiment that's shown in teal. So in teal, you've added the TRPV4 agonist, and at 5 minutes, you've added an antagonist. Well, we obviously haven't adversely affected the cells because the conductance goes back almost to normal levels. So the cells return to their baseline level. So there is the conductance issue. There's also, let me return to the short circuit current. Because in the short circuit current, we say, well, that whole thing is over in 5 minutes. How interesting can it be? Well, I think we have to consider that if the conductance is that high, the whole response is not over. It is, we think that what is going on is we have at least 2, if not 3, different transport phenomenon going on. And in those transport phenomenon, we have positive and negative charges moving in the same direction, which is why they seem to cancel each other out. Okay, I'm going to skip this slide in the interest of time and just come to this to say, well, if all of this movement across the cells of electrolytes is happening, is it accompanied by fluid flux? And the answer to that is yes. We are actually able to measure the fluid flux across the transwells. So what you're looking at is the flux in 10 minutes, the amount of fluid produced in 10 minutes across a transwell. In the control, which has diluent only, there's no statistical change from 0. In the TRPV4 agonist-treated cells, you see that there is a statistically significant increase in fluid movement, and that fluid movement is in a secretory direction, in other words, into the CSF side of the cells. That fluid movement is blocked by the pre-addition of a TRPV4 antagonist. So now we come to our working hypothesis as to what could possibly be going on in these cells. TRPV4 allows calcium to come in, it allows sodium to come in. The calcium can secondarily affect calcium-activated potassium channels, and I will say we have done the PCR, and we found that all 3 of the calcium-activated potassium channels, BK, IK, and SK, are present in the cells. But by using effectors, we feel that only IK is involved in the fluid flux. There are also calcium-activated chloride channels, TMEM16A, and to a lesser degree CFTR. They are also present in the cells. We are also looking at those. As far as the sodium is concerned, the increase in sodium has the potential to change the NKCC1, the sodium-potassium 2 chloride transporter. It could potentially affect the pump or other sodium channels that may be present in the cell. We do not find the epithelial sodium channel in these cells. Other people have reported that it is in the choroid plexus. We haven't yet looked at the sodium hydrogen exchanger. The sodium potassium 2 chloride channel has been shown by other investigators to be very important in CSF production, but also quite controversial in the field. And the controversy stems to whether this is an efflux or influx channel. I don't have time to go into all that controversy. I will show you in a moment some data we have, but that is an open question yet. So coming to that controversial question, what about the NKCC? Well, while it is likely extremely important in CSF production, it does not appear to be a big player in TRPV4 activated ion or fluid movement. What we're doing here is adding in the teal and the pink lines, we've added 2 different concentrations of bumetanide, which is an inhibitor of the NKCC. These are rather industrial concentrations of bumetanide, so if NKCC was involved, we should see some effect if it was involved in the TRPV4 transport. When we add TRPV4 to all 3 of these experiments, we see that there's a normal upswing. The downswing, which is probably mediated by other transporters, may be slightly affected by the bumetanide. The conductance change is not at all affected by the bumetanide. The other thing I would very quickly like to go through is the fact that TRPV4 is activated by phosphorylation events, and we feel that there is a kinase involved. That kinase is serum glucocorticoid-induced kinase 1. This was synthesized and characterized by colleagues in Italy, Professors Sciannoni and Perotti. They were interested in looking at the SGK1 in renal cells, but were kind enough to share the inhibitor of SGK1, SI113, and we tested it in our human choroid plexus cell line. If you add the inhibitor of the SGK1, as is shown in the pink line, you block most of the TRPV4 increase in short circuit current and all of the TRPV4 increase in conductance. That too is reversible if you add a TRPV4 antagonist at time 0. So SI113 seems to block both TRPV4 stimulated ion transport, and it also, shown in panel C, blocks TRPV4 fluid flux across the membrane. So what does it do in animals? Well, it does exactly the same thing that TRPV4 does. In the affected animals, which are seen here, I apologize, I'm going through this rather rapidly. You see the increase in hydrocephalus in the affected animals that is completely blocked by the addition of the treatment with SI under the same conditions as the ones I showed you in the TRPV4 experiment. What I didn't show in the TRPV4, but is also on this, is that it also decreased the head sizes in the animals. So, in the interest of time, I'm going to skip the next 3 slides. They will be available. You can look at them if you wish when they're posted on the APS website. But one final thing that I would like to consider, and that is that hydrocephalus, coming back to the clinic, is a condition that has multiple pathologies and is going to affect many different cell types in the brain. So you're going to have inflammatory changes, neurological changes. We know that there are vision changes. So the only way really we feel to treat this disease is going to be combinatorial drug treatment, not only targeting the CSF production, but targeting inflammatory pathways as well as other pathological changes. And we are involved in some multi-institutional collaborative studies beginning to look at combinatorial drug treatments. So I do believe I'm out of time, so I'm going to stop at this point, and I would be happy to take any questions. So what I'm going to do is stop sharing my screen and put my camera back on.
Okay, great. First and foremost, thank you for that excellent talk. It was a pleasure to learn a little bit about a field that I'm not so familiar with, and I'm sure the audience can agree that that was quite informative and really nice to hear about. So I'll start off with the question and answer period. The first question we have is a very simple one that came earlier in your talk, and it's a question that says, if cerebrospinal fluid is not plasma filtrate, can we then say that cerebrospinal fluid is an ultrafiltrate of the plasma?
I think we're talking semantics there. I think what is important is that the composition of the CSF is different than the plasma, and that composition is carefully controlled and has long-ranging implications, everything from diurnal rhythms to amount, if you will. So my point there was that the control of the CSF resides in those epithelial cells, and that's why we're studying those cells. Thank you.
Thank you, Bonnie. Lots of reaction, positive reaction and appreciation. I don't know if you can see them yet, but
I can see the hand clapping.
Next question that also came earlier in your presentation. Could we now be certain about the direction of apical NKCC1?
That is a question that I do not have data to contribute to. Okay, which way is the NKCC moving? There are people that are studying that in depth. There are some really good studies, and it's interesting because in adult animals, the studies seem to indicate that if you block NKCC, you will decrease CSF production. In a neonatal model from a different lab, if you increase the NKCC, you decrease CSF production. Now, that may be a developmental issue. We're talking adult animals versus neonatal animals. I am unaware of any data showing that the NKCC1 knockout animals have differences in ventricle size. I'm quoting literature here because while it's something we have begun to look at, I don't think we have enough data for me to weigh in on that question yet. It's a very important question.
Thank you. I think we can continue.
Okay, great. The next question we have is whether you think that TMEM67 is directly impacting, the question just disappeared on me there, is directly impacting TRPV4 function, or that TRPV4 function is just a helpful way to modulate the hydrocephalus phenotype.
I think the latter. Now, our genetic model, we're well aware, is not the best model because it is mimicking a very rare disease. What we're doing is trying to develop better models that are more physiologically relevant. So I have preliminary data. It was data that was used to write a grant, and these data come from a collaboration with two professors at Johns Hopkins, Lauren Jensky and Dodie Robinson. We used their post-hemorrhagic model. So this is much more physiologically relevant. It's a post-hemorrhagic, also rat model, and we tested the TRPV4 agonists or antagonists in that model, and they were effective in that model as well. So I think from the question, it is the latter situation where TRPV4 is probably applicable to several forms of hydrocephalus. We haven't tested it yet in post-infectious hydrocephalus, but I think it's a stress protein that is activated probably by the increase in the cerebrospinal fluid and reacts to it. But that's theoretical.
Great, next question is about TRPV4 location. And the question is it located throughout the choroid plexi, lateral and post-ventricle? And is it absent along the ventricle surface?
If I'm understanding the question correctly, the question is it in all three different types of choroid plexus, the lateral, third, and fourth ventricle. The answer to that is we are not absolutely certain. We've looked at it in the lateral ventricle and the fourth ventricle, and it is there. It is on the apical surface, both by immunohistochemistry, which I did not show at all, and by our electrophysiological studies. It is the TRPV4 agonist is only effective from the apical side. If you add it to the basolateral side, you have no effect. So both of those things say that it is located on the apical membrane, not on the basolateral membrane.
Okay, outstanding. Due to the interest of time, unfortunately, we can't proceed with any more questions. There are a lot though. If you'd like, you can always reach out to Dr. Blazer-Yost if you have important questions pertaining to her work. I believe her email was flashed up on the screen at some point in the presentation. So when you go back, you can find that information as well. But on that note, we'd like to once again congratulate you on this distinguished award, and thank you again for this wonderful lecture. It was truly great to see the body of work throughout the years. And then also on that note, we'd like to thank all the attendees who took time out of their day and attended this webinar. We appreciate your time as well.
Thank you so much. Thank you.